Let's give the students another minute to log in, and then we'll begin. I've already got that downloaded, so I'm not sure why that's not working. Okay, let's begin. I've got it recording already. Oh, there we go. So we were talking about osmosis last time. And we finished talking about this slide here, showing you a hypertonic solution where water leaves the cell and the cytoplasm pulls away from the cell wall. This can lead to plasmolysis. Generally, most cells will die. If it's a endospore, of course, it will not die, but most other cells will die. Now, active transport is the movement of material against the concentration gradient, and it is used whenever the cell finds itself in a desert-like environment or a desert-like environment in the ocean where the nutrients, the concentration of nutrients, are very sparse. And what's the cell to do when it wants those molecules? Well, it pumps them against the concentration gradient. Cells accumulate the nutrients they need against a concentration gradient, meaning you're pumping it from an area of low concentration to an area of high concentration. This requires energy. The cell is performing work to move the molecule across the cell membrane. Cells can do this by active transport, where they actually have a protein pump moving the solute molecule against its concentration gradient. And it's a transport protein, or it could be called a transporter protein, in the cell membrane. Generally, they're molecule specific, so we have one for each molecule the cell wants to move into the cell. And for each molecule that moves in to the cell using the transport protein, the cell uses up 1 ATP, adenosine triphosphate. Any questions about any of that? Now, all cells have active transport, but only prokaryotes have group translocation. It's a special form of active transport found only in prokaryotes. It does require a transport protein to move the molecule across the cell membrane. It does require a high energy phosphate compound like PEP. And then the molecule that's transported in group translocation is chemically altered. Like glucose, if it's moved into the cell, it will be chemically altered to glucose phosphate or glucose 6-phosphate. And what happens is PEP transfers a phosphate group to the sugar. It's thought that the alteration of the molecule can prevent the escape of the molecule from the cell and allow the cell to accumulate substances at higher concentration than would otherwise be possible. For example, a prokaryotic cell that is trying to transport glucose into the cell, if there was no group translocation and there were 10 molecules, it would, using active transport, move all of those 10 molecules into the cell. So let's say they're glucose. Now the first one being brought in isn't a big deal, but the 9th and the 10th being brought in is a big deal because the concentration gradient is so great. And it's thought that with group translocation, you now don't have to bring in the number 9 and the 10 because you brought in 5 and then you brought in 5 by group translocation. And so when you transfer glucose into the cell, the concentration gradient is now less, 50% less. Because not only do we have glucose in the cell, we have glucose phosphate in the cell. Any question about any of that? Remember, group translocation is something that is only seen in prokaryotic cells, and it's a special active transport. Eukaryotes do not have group translocation. All right, let's briefly talk about the cytoplasm. That's the liquid inside the cell membrane. And because we're talking about bacteria, that's all of the liquid portion inside the cell. It's mostly water, about 80%, but it does have dissolved solutes in it. And everything else that is inside the cell is in the cytoplasm, at least of a prokaryotic cell. The nucleoid region is in the cytoplasm, the ribosomes, the inclusions, and these are regions of the cytoplasm that either stain differently or because the ribosomes are so big and the inclusions are so much accumulated stuff that it precipitates out of the cytoplasm. The ribosomes actually are so big they don't go into solution. Remember that the nucleoid region is just wherever the DNA is floating in the cytoplasm. I should point out that the chromosome, which is most of the DNA of the cell, is generally in a bacteria 1 molecule of DNA. But there are smaller molecules of DNA that we call plasmids. And how they differ is a plasmid just has a few genes on them. And then when we're talking about the chromosome, it has 20,000 to 100,000 genes on it. Any question about any of that? So prokaryotes do have a nuclear area. We call it the nucleoid. It has no distinction between the nucleoid region and the cytoplasm. There's no boundary. It's just wherever the DNA is floating in the cytoplasm. That is the nucleoid region. And it stains differently when we stain it and look at it under the microscope. Actually here it's even looking different under the electron microscope. Ribosomes are the site of protein synthesis, and they're made of both ribosomal proteins and ribosomal RNA. We'll talk a little bit more about ribosomal RNA a little bit later in the term. You don't need to know the makeup of ribosomes, but there is a lower subunit, smaller unit, and then an upper, larger unit. And together in prokaryotes, they make a ribosome of 70S in size. The S is what biochemists use to measure ribosomes, and is simply where they sediment in an ultracentrifuge gradient. And if you don't need to know that, but you do need to know that the ribosomes of prokaryotic cells are 70S. Those are smaller ribosomes than a eukaryotic ribosome. Eukaryotic ribosomes are 80S in size, and they are larger. I briefly discussed inclusions. Inclusions are deposits that the cell accumulates, and there's so much of it in one region that it precipitates out of solution, forming what we see as an inclusion under the light microscope. There can be many different types, and the cell generally uses these inclusions for resources. You don't need to know the different types. Just know that there's different types. And here we're looking at inclusions under the light microscope. This cell metabolizes sulfur, so it's accumulating sulfur in the cytoplasm, and so much sulfur gets there that it literally precipitates out of the cytoplasm. And then we see it as an inclusion. And there's a third one. Let's talk a little bit about endospores. I think we've talked about them already, but endospores are specialized resting cells that can survive harsh environments. They're mainly seen in the families of Bacillus and Clostridium. When the environment starts becoming detrimental to the survival of the parent cell produces an endospore. Note that this is not reproduction. One parent cell produces one endospore, and then the parent cell will die as the environment deteriorates. But the endospore can survive and will survive until the environment becomes more favorable. Endospores are highly resistant to nearly everything: desiccation, heat, chemicals, radiation, and generally they survive conditions that the vegetative cell cannot. Sporulation, or sporogenesis, is endospore formation, the forming of an endospore. And then when the environment becomes favorable, the endospore can undergo germination and return to the vegetative state, a metabolic cell without the endospore. Endospores have very little metabolism going on. Like I said, they're a survival structure. They're not a reproductive structure. They're not a metabolically active cell. I'm not saying that they have no metabolism, but their metabolism is greatly reduced. Let's look at endospore formation in a little greater detail. When the environment starts deteriorating, the parent cell divides the DNA like normal, And then the DNA moves towards opposite sides of the cell like normal. But then something different happens. Instead of the cell dividing equally down the middle, it divides into a smaller cell that will become the endospore, and the plasma membrane forms about it, and then the spore septum starts forming about it. A peptidoglycan layer forms between two membranes, so the endospore has two membranes. And then, oops, went too far. And then the spore coat forms around the endospore. The parent cell then dies because the environment has become unfavorable to survival, and that will release the endospore from the parent cell. Any question about any of that? Oh shoot, I'm not seeing my video working. Oh, there it is. So let's watch a little video on endospore formation. It's only 8 seconds long, but it's taking a while to load. And there's the endospore. Any questions? So remember, an endospore is a survival structure. It is not a reproductive structure. Any questions about bacteria and prokaryotes in general? If not, I'm going to move on, and we're going to go very rapidly talking about the cell structures in eukaryotic cells. Where I'm going to go very rapidly is supposedly you've had this in Biology 160. So you should have already had all of this material, and it should be reviewed to you. We're looking at, on this half of the cell, an animal cell, and the organelles you can find in an animal cell. And on this half of the cell, a plant cell, and the organelles you can find in a plant cell. Most of the organelles you find in both plant and animals, like mitochondria, Golgi apparatus, endoplasmic reticulum. But there are some that you only find in one. For example, you can find a vacuole in an animal cell and a vacuole in a plant cell, but you only find the central vacuole in a plant cell. And that's probably what they're showing you there. The central vacuole is the largest organelle, membrane-bound organelle, in a plant cell. In an animal cell, the largest membrane-bound organelle is the nucleus. Vacuoles tend to be much smaller than the central vacuole. You can find chloroplasts in plant cells. You never find them in animal cells. Okay? Any question about any of that? And here we're looking at the same thing, only with an electron microscopic image. Let's talk a little bit about a comparison of the prokaryotes and the eukaryotes. Let me see if I can blow this up. Come on. That's better. Move that over here. So the size of a prokaryotic cell is smaller than a eukaryotic cell. And actually 0.02 is a little too small. That's probably a non-viable cell. But the general size is 0.04 micrometers to 2, actually up to 10 micrometers, the largest prokaryotic cell. Generally, prokaryotes are 1/10 the size of a eukaryote cell. Generally, eukaryotic cells are 10 micrometers to 100 micrometers in diameter. And as you know, the yolk of a chicken egg cell is much bigger than 100 micrometers. There is no nucleus in a prokaryotic cell. There is a nucleus in a eukaryotic cell. Membrane-bound organelles are absent in prokaryotic cells. They are present in eukaryotic cells. Flagella are present in prokaryotic cells and in eukaryotic cells, but they differ, and that is the flagella arrangement is more complex in eukaryotes than in prokaryotes. The glycocalyx can be present in prokaryotic cells. Not all cells have them, but some do. And they can be as a capsule or a slime layer. In eukaryotes, the glycocalyx can be present in some cells that lack a cell wall. The cell wall is usually present in a prokaryotic cell. It's chemically complex. It includes mainly peptidoglycan in the bacteria. The cell wall of the eukaryotic cells is chemically simple. Like in green plants, the cell wall is simply made up of cellulose. Okay? And then animal cells, of course, don't have a cell wall. The plasma membrane is actually very similar between eukaryotes and bacteria. It's a lipid bilayer, phospholipid bilayer. There are some differences that the bacteria tend not to have carbohydrates, and most of them lack sterols in the plasma membrane. And then animal cells have sterols in the plasma membrane to give it strength. And we tend to have carbohydrates that serve as receptors on our cell membrane. The cytoplasm, this is a little bit out of date, this table, and I do not know why the author has not changed that. It is known that prokaryotes do have a simple cytoskeleton. They don't have any cytoplasmic streaming. And then eukaryotes have a complex cytoskeleton, and they have cytoplasmic streaming. We'll take a look at what cytoplasmic streaming is. It's the movement of the cytoplasm around the cell. Any question about any of that? So yes, prokaryotes do have a simple cytoskeleton, and then eukaryotes have a complex cytoskeleton. I already talked about this one. The ribosomes of prokaryotes are smaller than the ribosomes of eukaryotes. And I do need to mention that there are organelles, the mitochondria and the chloroplasts, that do have smaller 70S ribosomes. And we'll talk a little bit about that later in this lesson. The chromosomes are different. Generally, bacteria have a single circular chromosome that lacks histones. And then eukaryotes generally have multiple linear chromosomes that contain histones. The cells divide differently. Bacteria generally divide by binary fission, where one cell literally splits into two. And eukaryotic cells do have cell division, but we go through cell division through mitosis. And then sexual recombination, it's generally absent in the prokaryotic cells. They can transfer DNA, usually plasmids, from one cell to another. Sexual recombination does happen in eukaryotic cells, and it involves meiosis. All right, any question about any of this? If not, let's move on. Eukaryotes can move by flagella, and this is the only way that prokaryotic cells can move. Eukaryote cells can also move by cilia. Flagella is a long whip-like structure. Cilia are shorter hair-like structures, and usually there's few flagella on a cell. Even if the cell has many of them, it's not as many as the cilia, meaning, the peritrochus arrangement of flagella. It's still under 20, for example, and the cilia, usually it's hundreds of them. So, few and long flagella, many and short cilia. They're both used for locomotion of eukaryotic cells. And I should say that eukaryotic cells have a third way of moving, and that is with pseudopods. Any question about any of that? The cell wall is present in plants, in algae and fungi, but the cell walls are chemically simple. They have no peptidoglycan. You don't need to know about the makeup of the cell wall. I did mention that plants have a cellulose makeup, and that one you probably already knew. Cell walls are not present in animal cells. They instead have a plasma membrane covered by a glycocalyx. And in their plasma membrane, they can have carbohydrates, usually bounded to proteins and lipids in the membrane. And we can use those as receptors. The cell membrane of eukaryotes is very similar to bacteria. And it is actually thought, we'll talk about that eukaryotes borrowed the cell membrane from bacteria. They found it efficient, so they borrowed it. So the cell membrane is mainly a phospholipid bilayer, but it does contain peripheral and integral proteins. These include transmembrane proteins, proteins that span the membrane. The cell membrane of eukaryotes is selectively permeable, the same as it is in prokaryotes. And to transport material across the membrane, eukaryotes can use simple diffusion, and with oxygen and carbon dioxide, that's what we use. We can use facilitated diffusion, and I did mention before that the best example is with fructose. It moves into the cell by facilitated diffusion, moving from an area of high concentration to an area of low concentration. But it does use a transport protein. This transport protein does not spend any energy, so no ATP is used. And that is why when you eat fructose, you'll never digest all of it because it'll only move into the cell to the point where the concentration outside the cell equals the concentration inside the cell. Osmosis refers to the movement of water across the eukaryotic cell membrane. And then cells can move material into the cell or out of the cell by active transport, moving a molecule against its concentration gradient. For example, when you eat sugar and it's glucose, your body will move every single last molecule of that glucose from your gut into your intestinal cells. And it'll do that by pumping the glucose against the concentration gradient from areas of low concentration to areas of high concentration by active transport using a protein transport molecule, active transport molecule, and spending ATP to move each molecule. Eukaryotes do not have group translocation. However, they do have vesicular transport, where vesicles can move material into the cell and out of the cell using a vesicle. Prokaryotes cannot use vesicles to move material around in a cell. For one thing, a vesicle is a membrane-bound organelle, and prokaryotic cells don't have them. There are two types of vesicular transport: endocytosis, the movement of material into the cell via a vesicle, and exocytosis, the movement of material out of the cell by a secretory vesicle. What happens is the cell makes something it wants to secrete, it puts it in a vesicle, it then moves the vesicle to the cell membrane. The phospholipids of the vesicle fuse to the cell membrane, and then they open up, and then the contents of the vesicle are spilled outside the cell, and the cell membrane is always kept intact because the vesicle moves up to the cell membrane, and then the membranes fuse. The material is then released. Any question about exocytosis? Endocytosis is the movement of material into the cell using a vesicle. There's two types. Phagocytosis, where the cell can put out pseudopods and then can extend the pseudopods and eventually surround what it wants to engulf and phagocytize. This is called cellular eating, phagocytosis. But it can also have endocytosis by pinocytosis, and that is something moves to the cell membrane, and then the cell membrane starts folding in, and then it just folds in and then forms a vesicle outside the cell membrane, and then the vesicle can move into the cell. This is called cellular drinking, pinocytosis. Any question about any of that? All right, the cytoplasm for a eukaryote is all of the aqueous substances inside the plasma membrane, but outside of the nucleus. The cytosol is the fluid portion of the cytoplasm. So, for example, the ribosomes are in the cytoplasm, but they're not dissolved in it, so they're not part of the cytosol. Any question about any of that? Cytoplasmic streaming is the movement of the cytoplasm around the cell that can move nutrients from one region of the cell to another. And I'm trying to show you a little video of that. And these videos take a long time to load, which is why I tried to get it started early.
Grammarly does more than catch errors.
Well, we got a commercial. Isn't that fun? So this is a real-time video showing you the chloroplast moving around the cell from cytoplasmic streaming. And there they're moving on that side of the cell. Oh, this is a much bigger picture and easier to see. So cytoplasmic streaming, where the cytoplasm is actually moving in the cell, and that moves nutrients from one region to another. In green plants, it's moving the chloroplasts as well, and that's for getting them better exposure to light. Any question about any of that? Cytoplasmic streaming is only found in eukaryotic cells. Let's talk about the cytoskeleton. It's the structural framework. You can consider it the cell's backbone. It consists of 3 types of protein filaments: microfilaments, intermediate filaments, and microtubules. And here is a picture of the cytoskeleton of a cell. And we don't see the exact cell, but you can sort of make out that it's right here. And this one, we're seeing so much, you can actually make out that this is the cell here. Now, that might even be another cell there, but this is the main one there. The point is that eukaryotes have a very complex cytoskeleton. Any question about the cytoskeleton? Its purpose is to give structure and support to the cell. One other thing it does is that it could anchor a protein if the protein is attached to the cytoskeleton. But protein is not going to move around in the cell. All right, let's move on to the ribosomes. It's the site of protein synthesis. In eukaryotes, the ribosomes are larger, 80S, and there are 2 types. The ribosomes can be membrane bound. That means they're attached to the rough ER, or they can be free, found in the cytoplasm. By the way, the reason why we have rough ER and smooth ER is smooth ER has no ribosomes attached to it, and rough ER has ribosomes attached to it, and that's why it looks rough. Any question about any of that? Although eukaryotes do have larger ribosomes, the 80S in size, they do have ribosomes that are at 70S in size. And these 70S ribosomes are only found in the chloroplasts and in the mitochondria. Any question about that? So yes, eukaryotes do have 80S ribosomes, but the chloroplasts and the mitochondria have 70S ribosomes. And you're just going to have to remember that. Although I will talk to you about a theory that'll help you remember that. All right, eukaryotes have organelles. By organelles, I mean specialized membrane-bound structures. They allow compartmentalization of the different functions in a cell. Each organelle has a characteristic shape and a characteristic function. They generally perform one specific function, like the mitochondria. Its function is to convert glucose into ATP, and their functions can be involved in cellular growth, maintenance, and reproduction. Any question about any of that? Here are the different organelles. We're going to talk about most of them, but not all of them. The nucleus, its function is to contain and protect the DNA, and you could say chromosomes instead of DNA. The endoplasmic reticulum, there are two types, the smooth ER, which is generally making lipids, and the rough ER, which has attached ribosomes, which is generally making proteins. The Golgi complex, it modifies and sorts and packages the proteins that are made by the rough ER, and then the proteins are sent to the Golgi complex for modification. Okay? Any question about that? We'll talk briefly about that, a little more detail in a little bit. Lysosomes are vesicles for digesting things, and they have a bunch of digestive enzymes in them, including lysozyme, which gives the vesicle its name, lysosome. Any questions about that? So a vesicle, which I haven't really talked about here, so I should talk about it. A vesicle is a membrane-bound organelle that moves material from one position in the cell to another. Once the vesicle stops moving, we can call it a vacuole, because now it's no longer moving, transporting material around, it's storing material. And we call vesicles that simply store material a vacuole. Vacuoles and vesicles, when we look at them under the microscope, look the same. And like I said, initially a vesicle can be moving, and we call it a vesicle, and then it stops moving and starts storing, and then we call it a vacuole. Any question about any of that? Animal cells do have vacuoles, but only plant cells have a central vacuole. In plants, the central vacuole is the largest organelle in the cell. And the purpose of the central vacuole is to store material, both waste and nutrients. And that's just the way plants are. Imagine storing your waste and your food in your refrigerator. That's what green plants do in the central vacuole. Okay? All right. The mitochondrion, I think I've already talked about it. It's the site of ATP synthesis and cellular respiration. You can think of it as a cellular powerhouse, how the cell makes energy. Chloroplasts are only found in plants, and they're the site of photosynthesis. I should say they're not found in animals. You can find them in algae, well, that would be it. Algae and plants. They're the site of photosynthesis. Perioxosomes, it's a special vesicle that contains various enzymes for the oxidation of organic substances. And it can destroy things by using H2O2, hydrogen peroxide. And that's why we call it a perioxosome. The centrosome is involved in cell division. That's where we have the centrioles, which is involved in forming the mitotic spindle. The Golgi complex modifies, sorts, and packages proteins received from the endoplasmic reticulum and then transports them to different destinations. For example, the Golgi complex could package the proteins, modify them and package them, and then send them for extracellular secretion, releasing the hormones, releasing the proteins, from the cell. You can think of that maybe in a teenager when they're going through their teenage fits. They're releasing a lot of extracellular proteins. And what this Golgi apparatus does, it sends it to the proteins to the plasma membrane. How it transports the proteins around is by using vesicles. And actually that's how the endoplasmic reticulum makes the proteins, the rough ER, and then sends it to the Golgi apparatus with a vesicle. The vesicle fuses on this side of the Golgi apparatus, and then the protein will be modified by the Golgi apparatus, and then on that side of the Golgi apparatus, the secretory vesicles will come out, a vesicle will be made, and then the protein will go to another location in the cell by the vesicle. Any question about that? Okay, I guess we're good, so we're moving on. So the eukaryotes have an endomembrane system, and that's where various organelles are part of a cellular transport system. And they move things around from one place in the cell to another using vesicles. And that is called the endomembrane system. Trying to get this to load.
This ocean.
We're really getting ads
Welcome
today.
to Moo Moo Math and Science. In this video, I'd like to talk about the endomembrane system. The endomembrane system consists of organelles that create, modify, and transport proteins and lipids throughout the cell. It consists of the nuclear membrane, rough and smooth ER, the Golgi apparatus, and different types of vesicles. First, ribosomes, which are protein factories of the cell, can be found on the rough ER. These ribosomes create proteins and then transfer these proteins into the ER, where they undergo modifications, such as folding. These modified proteins may be absorbed into the membrane of the ER or packaged into a vesicle that may travel to the Golgi apparatus. The smooth ER may transport lipids to the Golgi apparatus, also using vesicles. A vesicle is a relatively small membrane-bound enclosed sac that stores or transports substances. At the Golgi apparatus, the proteins are sorted, packaged, and tagged so that they can travel to the right place inside or out of the cell. The receiving side of the Golgi apparatus is called the cis face. The opposite side is called the trans face. The transport vesicles that formed at the ER travel to the cis face, fuse with it, and empty their contents into the Golgi apparatus. As the proteins and lipids travel through the Golgi apparatus, they undergo further modifications that allow them to be sorted. The most frequent modification is the addition of short chains of sugar molecules. These newly modified proteins and lipids are then tagged. These tags help the proteins arrive at their proper destinations. Finally, the modified and tagged proteins are packaged into vesicles that leave the transface of the Golgi apparatus. Some of these vesicles deposit their contents into other parts of the cell where they may be used, while others release their contents outside of the cell by diffusing into the cell membrane. So there we go, the endomembrane system. Thanks for watching, and Moo Math uploads a new math and science video every day. Please.
Okay, any questions? So this picture shows you the endomembrane system, which we already discussed it in the video, so I'm not going to go over it. So a vacuole, a vesicle, and a lysosome all look pretty much the same in a cell, and their only difference is the function. A vesicle is moving something from one area of the cell to another. A vacuole is storing something in the cell, and then a lysosome is a special vesicle for digesting things. All right, let's talk a little bit about the mitochondrion. It's the site of cellular respiration, where the cell, at least eukaryotes, make ATP synthesis. So it's the cell's powerhouse. It has two membranes, an outer membrane shown here, and then an inner membrane which is folded, greatly folded on itself, which is the inner membrane. And the folding is to increase the surface area. So when we're looking at it under the electron microscope, here we're seeing the folding of the inner membrane. When we look in the mitochondria, we find that it has a DNA molecule in it and ribosomes. But the ribosomes are not 80S in size like the majority of eukaryotic ribosomes. They're 70S in size. Any question about that? We'll talk more about cellular respiration in a later lesson. The chloroplasts are organelles found only in green plants and in photosynthetic algae. It too has two membranes, an outer membrane shown here, and then an inner membrane which is greatly folded on itself, showing you the granum here. And the flattened membrane stacks we call thylakoids, and they're arranged into stacks called grana. And there's a granum right there. When we look at the chloroplasts, we find they have a DNA molecule in them, which we don't see it here, or even here. But it does have a DNA molecule, and it has ribosomes in it. These ribosomes are also 70S in size, smaller than the eukaryotic 80S size. Any question about that? If not, let's talk about why people think that eukaryotes have a mitochondria and a chloroplast, which have two membranes, a DNA molecule, and of course ribosomes. Come on. All right, a long time ago, when there weren't eukaryotic cells, there were proto-eukaryotic cells. The proto-eukaryotic cell, I'm just going to abbreviate that PE, consumed a bacteria. And then, for reasons unknown, the PE cell did not eat the bacteria. At least right away. Are you guys able to see this okay? Do you want me to blow this up? Hello? I'm going to assume you guys can see this okay. All right. Now, I can't draw that, but let's have a cell here, and it's a big proto-eukaryotic cell, and inside it we have a smaller bacteria cell. The bacteria could perform aerobic respiration. So the bacteria inside of the proto-eukaryotic cell took 1 glucose from the cytoplasm of the proto-eukaryotic cell and then made well, up to 38 ATP molecules. This is the bacteria that could do this. Now the PE cell, the proto-eukaryotic cell, can only make from 1 glucose, oops, wrong one. Can only make 2 ATP per molecule of glucose using fermentation. Okay? Any questions about any of that? Now, for whatever reason, probably because life was young, the bacteria released some ATP to the cytoplasm of the PE cell, the proto-eukaryote. This greatly confused the PE cell, because the proto-eukaryote lost one glucose molecule, but instead of gaining 2 ATP, it gained a large number of ATP. And we don't know exactly how many ATP it gained, because that happened a long time ago, but it's someplace between 2 and 38. Okay? So, the PE said, I am going to keep that bacteria, and he will make me ATP. Okay, any questions about any of this? And so the two lived together in symbiosis. The bacteria found a good home. It got nutrients from the PE cell, especially glucose, and then it made ATP with aerobic respiration and released at least some of them to the proto-eukaryotic cell. Okay, so the bacteria got a home and got glucose and then made ATP for the ATP for the proto-eukaryotic cell. And then that's the advantage that the proto-eukaryote got. It's getting more ATP from glucose than it could ever make from glucose. And so the two lived together in symbiosis for a long time. Now, we never find that bacteria outside of a eukaryotic cell. The two have lived along together for so long that we don't find the bacteria living on its own in the wild. And then we don't find, we'll put that in quotes because you could find an example where I'm wrong, but we're not going to get into it. We don't find a eukaryotic cell without having a bacteria in it. And what this bacteria has become our mitochondria. Okay, any questions about any of that? Let's look at the evidence for what I've just talked about. Where the heck do I have it? There it is. Well, in the first place, the endosymbiotic theory is that there are eukaryotes which engulf a bacteria, and then the bacteria serves or lives in symbiosis with the protostiston host cell, and then they live together for the benefit of both. The bacterium, in this case, engages in photosynthesis and then releases glucose to the protist, and the protist gives the bacterium a home. We have amoeba, which take up a bacteria, and then the bacteria engage in aerobic respiration for the amoeba, and these amoeba actually lack the mitochondria, and that's why they take up the bacteria to engage in aerobic respiration. And then the bacteria take glucose from the amoeba and then convert it to ATP and then release ATP back to the amoeba. Some evidence for this theory. First of all, the mitochondria is surrounded by two membranes. And if you think about it, everything that's brought into the cell will form a vesicle to be brought in. So it should have two membranes. The membrane of the cell and then the membrane of the vesicle. That's a weak part of evidence. Let's look at some stronger evidence. The DNA for eukaryotes is linear. The DNA for bacteria is circular and without histones for bacteria. The DNA for the mitochondria is circular and without histones. When we look at the first protein that's made in the start sequence from messenger RNA, it'll be methionine for eukaryotic cells. But for prokaryotic cells, it's a slightly different amino acid. It's formylmethionine. For the mitochondria, it's formylmethionine. Eukaryotes have an 80S size ribosome. Bacteria have a 70S ribosome. Well, mitochondria have a 70S ribosome. Okay? And then the greatest strength, when we sequence the DNA of the eukaryotes, of course they fall within the domain eukaryotes. When we sequence the domain of a bacteria, the DNA has the sequence of the domain bacteria. When we sequence the DNA of a mitochondria, where does it fall? Anyone guess?
Bacteria.
Yeah, it falls within the domain bacteria. Okay? And the chloroplast also, when we sequence the DNA of the chloroplast, it falls within the domain bacteria. So it looks like the theory I gave you, called the endosymbiotic theory, did happen in animal cells one time, and that bacteria that was engulfed by the proto-eukaryotic cell became our mitochondria. And nowadays, we never find that bacteria outside of a eukaryotic cell. The strange thing is that this happened more than once. And that is in with plants. Where do we have that shown here? Right here. First, they took in the mitochondria, and then they took in another bacteria, which became the chloroplast, and the chloroplast was a bacteria capable of photosynthesis, and that is why green plants can engage in photosynthesis, because they have in their cytoplasm a cell that used to be a bacteria, which has now become the chloroplast of the cell. Any question about any of that? Why am I off of this place right here? All right, if there's no questions, that's my last slide for today. We have discussed the endosymbiotic theory, how eukaryotes took up a bacteria cell and then started living in symbiosis with it. And that is how animal cells got their mitochondria, and how plant cells got their mitochondria and their chloroplast. I will see you at 6:30 for the lab. enjoy a little longer break today.
Thank you.
All right.